Yes, sir. You are presenting something, sir, or some other person, sir?
No, I am presenting.
Yes, I make you as a presenter, sir. You just
Okay.
do that, sir.
Okay. So how can I share now?
Yes, sir. The third icon is there, no, sir?
So I have mute, start video. Share is not still working.
Yes, sir. Yes, sir. Yes. Yes, sir. Yes. Could you try once? Could you try once, sir? I already make you as a presenter.
Maybe I will have to quit. Maybe I'll have to quit. Wait, I'll rejoin.
How can I share now? Sir, the third icon is there. Start video. Yes, we can able to see you, sir.
Ashish?
So, Professor Murthy Tenmalar here.
Tenmalar, how long is going to be the talk?
I don't know. I can ask Dr. Ashish.
Yeah, Professor Murthy, I'll just take maybe 40 minutes or so.
Okay.
Sure, absolutely. Absolutely. Yeah, as I understand. So I have tried to keep it. Perfect. Even they have
Thanks to both of you for making this happen. I think somebody has to mute everyone.
Yeah.
Lakshman?
Office actually. Office actually.
You've been seen on
Professor Murthy, shall I begin? Before we begin.
She can invite you to
Yes.
start.
So, good evening, everybody. Department of Biotechnology, I welcome you all for the webinar. And today's speaker is Dr. Ashish Mishra. Ashish Mishra did his PhD in IIC Bangalore. He did postdoctoral research in the Massachusetts School of Medical School. And he subsequently joined in IIT Hyderabad as assistant professor. And today he is going to talk about genetic coding, rewriting the genetic code using CRISPR technology. And it's a slideshow. You can clearly see that in two female scientists. One is Jennifer Doudna, another one is Emmanuel Charpentier, and another important thing is World CRISPR Day is coming on October 20th. So, considering all these things, we arranged this seminar. And without taking much time, I would like to invite Dr. Ashish to start his presentation. Thank you.
Thank you, Dr. Tenmalar, and thank you for this wonderful opportunity. I think I'm humbled and honored to talk about the work that led to Nobel Prize this year in chemistry. Couple of things that have been very, very remarkable about this Nobel Prize is that this discovery has been less than a decade old. So it is just 8 years since since the time we knew that about this machinery doing or being able to rewrite the genetic code to its getting the Nobel Prize. Raghu, can we mute the background? Okay. So 10 years back, nobody thought that this technology could have such a profound impact. And two amazing women scientists, as Tenmalar said, who got the Nobel Prize are Jennifer Doudna and Emmanuel Charpentier. Jennifer Doudna is a Chancellor's Chair Professor at UC Berkeley. She's also the Director of Innovative Genomics Initiative at UC Berkeley and also a Howard Hughes Medical Institute professor. Emmanuel Charpentier, when she began her journey, she began at University of Umeå in Sweden, but now she's a director at Max Planck Unit for Science of Pathogens. The summarize is that they found or they discovered a class of enzymes that are called CRISPR-Cas9 enzymes that can be tuned to rewrite and cut any piece of DNA in the way you want to. So this pretty much summarizes what they have done. Okay, so when this discovery was made, it made numerous headlines in newspapers and Times Magazine, and this is just one pictorial illustration of how well perceived this technology was and how much curiosity it generated in the time it was discovered. So right from making the cover page of Economist to going into MIT Technology Reviews to Nature, Science, Nat Geo, you think of the magazine and journal and they made it. And Jennifer Doudna was also named as Time's 100 most influential people last year. So the point I want to try and relay is that this journey from the discovery of tunable CRISPR-Cas9 system to date has been remarkable for both Jennifer Doudna and Emmanuel Charpentier. And apart from them, there are numerous scientists around the world who are using these genetic tools for various scientific purposes, right from doing basic research to innovation in agriculture to biomedical benefits of therapeutic usages. Okay, so if we look at the history of the CRISPR-Cas9 system, we know about the CRISPR system way back in 1987. So it was first discovered in E. coli in 1987. For a very long time, people did not know the role of this. So it took time till early 2000 when people started associating these Cas genes with DNA repeats in prokaryotes. And it's that people started realizing that CRISPR genes are something that can provide resistance to bacteria against viral infection. And in 2012, the science paper showed at the bottom is, which you can see through my cursor, is the one that led to the Nobel Prize, where lab of Jennifer Doudna and Emmanuel Charpentier together in vitro showed that CRISPR-Cas9 system can be used as a genome engineering tool. That means it can be used to engineer the DNA or program the DNA in a fashion that the experimentalist wants to. Okay, so very early on, the work done by numerous labs had shown that the CRISPR system was basically a mechanism for the bacteria to acquire adaptive immunity against viral infection. So this, for those of you who are not biologists or do not come from a biology background, this is a bacterial cell shown here. And on the top of this bacterial cell, you can see this. This is a virus or a phage that infects the bacteria. What happens when the bacteria infects the phage is the phage will leave its genetic material inside the bacteria. Whether the virus is infecting a bacteria or the humans, the process by which it does the infection is similar. That means whether it's infecting the bacteria or humans, it is going to put its genetic material into the cytosol. And what that genetic material will do is it will begin a molecular program that will have all the information for it to raise molecules or start a molecular program that will similar phage molecules inside the cells. And it will keep on repeatedly making viral particles till it kills the cell. The rule is simple. Now, what the CRISPR locus allows it to certain bacteria to do is that the bacteria in which the CRISPR locus is present, the genetic material once inserted inside the bacterial cell is fragmented into pieces and inserted in the CRISPR locus. So you can see that insertion as red, purple, yellow, and green blocks in this CRISPR locus right here. And what happens is this insertion in the CRISPR locus serves as a molecular memory for the viral DNA infection that has occurred in the bacteria. And this memory will keep on growing as the bacteria will encounter different kinds of infecting it. What will happen is each virus will inject its DNA into the CRISPR locus. Now, according to central dogma of molecular biology, the DNA is transcribed into a piece of RNA, and so is the CRISPR locus. So what the CRISPR DNA will do, it will be transcribed into a RNA. As you can see it in the form of what we have written as pre-CRISPR RNA. So these will be little wiggly pieces that you can see here, followed by this hairpin loop structure, and I will come to the hairpin loop structure in one of my later slides. But these pre-CRISPR RNAs will then be processed by certain processing enzymes that are present inside the cytosol of the bacteria, leading to individual CRISPR RNAs, or what we call as the guide RNAs. And these guide RNAs will get incorporated with the proteins that are made from these Cas genes over here to form something that we call as the CRISPR-Cas9 complex. 9 complex will have is elements or certain RNA elements that are complementary to the viral DNA that had already infected, as you can see in the orange, the red, the green, and the purple. And this does this is now a molecular memory that the bacteria recognizes that this viral particle has infected me earlier. Now I have made these CRISPR RNAs against it. So next time when it is going to infect me, these CRISPR RNAs, along with the CRISPR-Cas9 enzyme, are going to bind to the genetic material that the virus is going to insert inside them, thereby degrading the genetic material and not allowing the virus to multiply inside the host. This is how the CRISPR system was first discovered, and it was thought to be an adaptive immune response against an antiviral immunity in bacterial cells. This cartoon is rather a different form of the same illustration. However, what I want to convey from this cartoon is that this process is not only confined to the viral DNA that comes inside the bacteria. It can be done for any piece of external DNA that either comes inside the bacteria naturally or is inserted into the bacteria through an experimental system. Okay, so as you can see over here, this is a viral DNA that I talked about in the last slide, and this is a plasmid DNA, an artificial piece of DNA that you can insert inside the bacteria. The point being, what will happen is certain parts of this DNA are going to be inserted into the CRISPR locus, much like the viral DNA. This will again serve as a molecular memory for the bacteria. Once it's transcribed into the RNA, now those CRISPR RNAs will have certain molecular signatures of this blue plasmid DNA over here. And next time when this plasmid DNA would be put into the bacteria, it is going to recognize it as a molecular memory, and CRISPR-Cas9 complex will go and cleave it. That's how the process of adaptive immunity, or CRISPR, was originally hypothesized and found to work in the bacterial system. Now, what Jennifer Doudna and Emmanuel Charpentier did was picked the lead from here. So Jennifer Doudna was a biochemist, or rather an RNA biologist, which happens to overlap with the field I work on. And Emmanuel Charpentier was a microbiologist. So what Emmanuel found was that she found certain enzymes, which we later went on to know as Cas enzymes, in certain bacteria that she isolated in vials, that she found could cleave viral DNA in those bacteria. What Emmanuel's lab was, what Jennifer's lab was interested in was how these pre-CRISPR RNAs are made from the CRISPR array, how they are cleaved, and how they are incorporated in the Cas enzyme, and subsequently how this Cas enzyme and CRISPR RNA complex goes and recognizes the foreign DNA that enters into the host the second time and cleaves it. So this is where their collaboration started. Emmanuel, being a microbiologist, and Jennifer, being a biochemist, came together to find out what these proteins and RNAs do inside the cell. And that's where the real role of CRISPR-Cas9 as a programmable DNA RNA-guided DNA endonuclease came into picture. So if we look at the CRISPR-Cas9 system, it is nothing but RNA-guided DNA cleaving enzyme. Okay, so CRISPR stands for clustered regularly interspaced short palindromic repeats. That's what we call as CRISPR. And Cas9 is nothing but a CRISPR-associated nuclease 9, or what we call as Cas9. There are two sets of molecules that are very, very critical for the CRISPR-Cas9 system as a whole to function. One is the CRISPR RNA that I showed you in the last slide that comes from the foreign DNA that is inserted in the CRISPR locus. That comes with a short hairpin structured RNA called the tracer RNA to form something as a composite structure, what we know as the guide RNA. And then the nuclease enzyme or the CRISPR or the Cas9 enzyme, which is nothing but an RNA-guided DNA endonuclease. So what, if I explain in very simple terms, it does is the guide RNA goes on to find complementarity with the foreign DNA. And wherever it finds complementarity, it kind of comes together, as you can see over here. And then what the Cas9 nucleus does is, wherever the complementarity is found, in a specific manner, it will go on to cleave both strands of DNA, generating a nick. Okay, so this is more about a simplified way of understanding what the CRISPR-Cas9 system does. So what is the guide RNA? The guide RNA is nothing but a piece of RNA. It's a piece of RNA that directs Cas9 protein to the target site. And as I said, you can see in this picture over here, it has a target-specific CRISPR RNA that is complementary to the viral DNA that comes in or the foreign DNA that comes in. And then the tracer RNA, which keeps it in place for the Cas9 enzyme to do the recognition and for the cleavage. So, and this CRISPR RNA plus the tracer RNA or track RNA, what I have written, go on to together form the guide RNA. CRISPR RNA is the one showed in red over here is nothing but a 20 nucleotide sequence that is complementary to the target region in your gene. As I earlier showed in case of foreign DNA, I showed the viral DNA is complementary to the CRISPR RNA. And then the tracer RNA is nothing but something that provides a scaffold for the CRISPR RNA to bind and stabilize for Cas9 to be able to carry out its activity. Okay. Then comes the next part, that is the Cas9. Cas9 is the protein that is responsible for locating the DNA that has to be cleaved and cleaving it. This is applicable both in natural system, that means the bacteria, and the artificial system that Jennifer Doudna and Emmanuel Charpentier used in vitro to cleave a piece of DNA that they found interesting, which might have been or might not have been present in the bacterial system. So they took out these enzymes and guide RNAs in vitro and looked at cleavage of a particular piece of DNA. If you look at the Cas9 protein, it largely is organized into different, what we call in biochemistry or protein biology as domains. And it has 6 distinct domains. You can see the crystal structure on the right and a more cartoonic structure on the left. The domains are very well organized. The REC1 domain, REC2 domain, bridge helix, PAM interacting helix, HNH, and RUBC. So as I told you, Cas9 protein has 2 jobs. One, it has to locate the piece of DNA where it has to bind complementarily to the DNA and then go on to cleave it. So cleavage function is carried out by the RUBC nuclease and the HNH nuclease. HNH nuclease will cleave the piece, the strand of DNA that is complementary to the guide RNA, and RUBC will cleave the strand of DNA that is non-complementary to the guide RNA. The REC1 helix is the one that is responsible for the placement of the guide RNA at the right spot for the cleavage to occur. The bridge helix The one that you see in purple over here is the one that is responsible for initiating the cleavage reaction. And there's something called the PAM interacting motif that you see at the bottom. This plays a very critical role in deciding where the Cas9 enzyme is going to cleave. And I'll try to explain it in a little greater detail in the next slide. So protospacer, next slide. Over here, the PAM interacting motif, or the one that I talked over here as PAM. So what that PAM interacting motif, adjacent motif or protospacer adjacent motif does is, once the CRISPR RNA, tracer RNA, Cas9 complex is formed, what it does, it binds to the DNA and traces through the DNA till it finds a region that is complementary to the guide RNA. Now, one can assume that DNA being 3 billion base pairs, that there can be many regions that might be complementary to a particular guide RNA, because the guide RNA is only 20 nucleotides long. But there is one more catch. For the Cas9 enzyme to cleave, it has to find the PAM sequence. So if there is a guide RNA motif that, if the guide RNA binds to a DNA motif that does not contain a downstream PAM motif, and in case of Cas9, that PAM motif is called NGG, it is not going to cleave. The Cas9 enzyme is going to rapidly detach off if it does not find a proper PAM sequence. Only when it finds a proper PAM sequence along with the guide RNA complementarity is when it is going to cleave. And always the cleavage is going to occur 3 to 4 nucleotides upstream of the PAM sequence. Okay, so that's one usage of PAM sequence. The other and more important and physiological use of PAM is that PAM is the component that allows the CRISPR-Cas system to distinguish the bacteria's own DNA, means bacterial self DNA, from the non-self or the foreign DNA. Because it is only the foreign DNA that is going to contain the PAM sequence. And only when the PAM sequence is present is when Cas9 is going to cleave. For the bacteria's own DNA or the self DNA, there will not be a PAM sequence, and hence there will be no cleavage. Okay, so as I have written over here, PAM is an essential targeting component which distinguishes bacteria self from non-self DNA, thereby preventing CRISPR locus from being targeted and destroyed by the CRISPR-associated nuclease. Okay, so this is about 3 large-scale motifs that are present in the CRISPR-Cas system, and all the 3 components of this CRISPR-Cas molecular complex are tunable, and they are tunable in the way you want to. Okay. So the next step is, so now we have assembled the CRISPR-Cas9 complex. The CRISPR-Cas9 complex has found the PAM sequence, so it is going to cleave. Now, once the DNA is cleaved and the CRISPR and the cleavage point is generated, what is next? So then, because both the strands of the DNA are broken, the cell is going to employ something we call as a double-strand break repair mechanism inside the cell, which is illustrated very nicely in this cartoon. So if you look at the top panel of the image, you can see that the double-stranded nick is created in the first part of the pictorial. Now, there can be 2 mechanisms by which these broken pieces of DNA can be joined. One joining mechanism is something we call as a non-homologous end joining, in which the DNA repair pathways within the cell will take care of joining these 2 pieces of DNA. Now, most often than not, what this cellular process of joining will do is, it will create some kind of a change than what was normally existing in that part of the DNA, or the chemical letter that was existing over here would most likely be changed. Now, as is shown in green over here, that is something we call as the indel mutation. Means it can be an insertion, it can be a deletion, or it can be a change from the original nucleotide that was present. In most cases, such an indel mutation leads to a what we call as a premature stop codon, in which case the protein that is formed is not a full length or a fully functional protein, but rather a truncated version of the protein. This is very, very helpful in cases where we want to create knockouts of particular genes, or we want to study functions of particular genes by removing their function from the cellular system. And it is very widely used in both basic research and applied research. We also use this part of the technology routinely in our lab. Then there is the second mechanism, what we call as the HDR. What happens in this is, after the cleavage takes place, if you have a piece of DNA of your own choice that you want to insert at that place, whether it's a particular nucleotide or a particular change that you want, a single nucleotide change that you want to place over there, or you want to place a whole chunk of DNA over there, what you can do is you can provide a donor DNA inside the cell. And what the donor DNA will do is it will go and recombine at the site of cleavage, hence generating a position which would be edited according to your choice. And that is why I have used the term rewriting. That means you can precisely write it the way you want. So you erase the information by introducing the cleavage, and then you wrote the information the way you want it. And this technology is beginning to find numerous applications, both in biomedicine, in agriculture, in public health, and some of which I'm going to talk in later slides. Okay. So if I look at what I showed you that the CRISPR component, CRISPR-Cas9 has three main players. All those players are tunable at will. By at will, I mean you can tune them the way you want. And I'll just briefly touch upon them. The first component is the guide RNA. It's like the one that provides the GPS coordinates from where you have to go. In those 3.2 billion bases of DNA that occurs into the human genome, where the CRISPR-Cas9 has to go and bind is the information that is going to come from the guide RNA. Then comes the enzyme Cas9 that is going to bind and cleave that piece of DNA. Cas9 is like a molecular scissor. It will go, it is a biological molecule that is going to cut the DNA. Now, this cutting is tunable, right? Till now, what I talked about was we were cutting both the strands of DNA. We can tune this and people have already done it, have tuned this Cas9 enzyme to either cut only one strand of DNA, that function is something we call as the nickase function, or they have made it a cleavage dead. That means it will just go, bind, and sit over there. And that has also found numerous implications, wherein you have to either silence a gene or activate a gene. You just allow the Cas9 to go and bind and sit over there and not cleave the piece of DNA. And the third major player that has emerged is the donor DNA that you provide in cases where you are looking for HDR. For the donor DNA, you can have a donor DNA that contains any sequence you want to insert, any sequence of your choice, any sequence of your length, any sequence of those 4 ATGC letters, any combination of the 4 ATGC letters you want to have, you can tune them accordingly. So that is what, and this is what has led to the Nobel Prize for Jennifer Doudna and Emmanuel Charpentier, that they showed isolating all these players in a test tube and doing a reaction that all the components are perfectly tunable at will. Okay, so this is what the technology or CRISPR-Cas9 does in a system. It evolved as a bacterial host defense mechanism for adaptive immunity against viral infections, and now can be tuned according to will to cleave any piece of DNA of your choice. Since the discovery of Cas9, now researchers have discovered many Cas enzymes that can cleave DNA in different way, different manner, cleaving single strand, double stranded, recognizing different PAM motifs for Cas9, as I showed NGG. There are now CRISPR enzymes such as CPF1A and many others that can go on to cleave at PAM sequences that are different than NGG. And that has been an active area of research. Okay, so since the discovery of tunable properties of CRISPR-Cas9 in 2012, I just want to show you the number of publications that started to emerge out of CRISPR-Cas9 system. So you can see so 2012 is when Emmanuel Charpentier and Jennifer Doudna discovered the system. And since then, the number of publications have been rapidly rising. And I was reading somewhere and they wrote that the original Charpentier and Doudna paper has been cited roughly 10,000 times by now. And they were comparing, and on an average, every 8 hours, that publication is cited once. Apart from the basic research value of the CRISPR-Cas9 technology or CRISPR-Cas technology in general, it has found a lot of profound impact for applied research, both in market and in the patent landscape. It is estimated that by 2025, the CRISPR-Cas genome editing market in itself would be around 5.3 billion. 100, if not more, startups in Boston, in California, in New York are already working on some other aspect of CRISPR system, whether it's on agriculture, in biomedicine, in basic research, so on and so forth. So, and MIT has been one of the institutions that probably is holding maximum number of patents on CRISPR-Cas9 technology as of now. Okay, so this was about what the research has been all about and what the technology has been all about. Now let's see, much like any system, the CRISPR-Cas9 system has also come up with lots and lots of opportunities. But as opportunities grow, so do challenges. But according to what I have seen, and I believe the opportunities are far more than the challenges that we have faced till date. Okay, so in the public health regimen, if we look at the opportunities that CRISPR-Cas has provided, there has been very, very interesting research on something we know as gene drives. So gene drives is a way by which you can have certain genes or certain genetic patterns inherited in a way that is much more than the normal probability of 50%. So if you have a normal inheritance, what will happen is that there is a 50% probability of inheritance of a particular trait. And so the altered gene is not going to spread to a large population in the end. So as you can see in the red over here, that it doesn't go on to spread in a massive population. However, what happens in gene drive is, if you're introducing very, very rapidly a trait that did not occur originally in the genome, and that is what the CRISPR system can allow, it will allow lateral inheritance to occur, which means that the gene after a certain stage will almost be always inherited, as you can see in the red mosquitoes shown over here. And this thing has found a great deal of interest in engineering mosquitoes. So this is something that has been kind of researched a lot in terms of mosquitoes. And what people have been interested in is, can we engineer mosquitoes so that they do not reproduce or they cannot pass on parasites that they carry, which otherwise they would spread around. And this has found tremendous amount of interest in people who are looking at vector-borne diseases such as Zika or dengue virus or malaria. So it has tremendous amount of public health impact globally, both today and moving forward. The other area where it has it is beginning to find tremendous amount of impact. And I think I and many other researchers around the globe think that probably would be the first place where CRISPR-Cas9 technology is actually going to show a lot of impact is in agriculture. And this is a pictorial that I got from a Cell paper where what researchers from Cold Spring Harbor National Laboratory have been able to do is they have been able to use CRISPR-Cas9 to make certain changes in tomato genome so that you get many fold more fruits out of a tomato plant than you would otherwise get. And with the research, they found that these tomatoes are genetically identical to the tomatoes they would have got otherwise. And I mean, this has led people to think that the kind of impact that this can have over the crop market, or imagine if we can get 3 times as much fruit from a single plant that would otherwise be needed 3 or more plants to get. You can minimize the water usage, you can minimize the usage of pesticide, you can minimize the uses of nutrients, and so on and so forth. And this is just one of the several examples. Things are being done in wheat, in rice, and so on and so forth. There are a number of agricultural applications that we have been able to see. However, with these opportunities, there have been challenges. And one of the challenges that has been there or has begun to come up recently is one of these studies that came from a lab in Penn State, where they engineered the mushrooms such that these mushrooms will never turn brown. So they will always remain white. And you can just imagine the amount of marketing impact this will have, that the mushrooms will always look fresh. And what happened was, or what ignited a large debate was that these mushrooms were not subjected to very tight regulations by the US FDA, and they were approved to be sold. The problem that came was, or the reason why tight US FDA regulations were not applied was because CRISPR-Cas9 technology was not classified as something that would create a genetically modified crop. So what they did was they used CRISPR-Cas9 to introduce a particular mutation in a gene locus in the mushroom, which did not allow them to go brown. It is different from genetically modified crops where we insert a particular piece of DNA so that a particular trait that we are looking for, like BT cotton or BT brinjal, is acquired. And that is why this did not catch the attention of the US FDA. However, later on, now moving forward, the regulations both in US, in India, and elsewhere have been tightened on CRISPR-Cas9, and now CRISPR-Cas9 based any changes in crops are classified as genetically modified crops and will be subjected to rigorous evaluations. And there was a study in Nature that showed how European Union dealt a blow to CRISPR crops by saying, okay, no, any changes that are done with CRISPR-Cas9 will classify the crop as a genetically modified crop, and so it has to go years of evaluation before it can be allowed to sold. Imagine if it would go unnoticed, it would have tremendous amount of impact on the market. So these are some of the challenges that have begun to arise. So now I want to shift a little bit and show some opportunities that have arisen in field of biomedicine. And one of the things that CRISPR has offered right out of the throat is point-of-care diagnostics. So what Jennifer Doudna's lab, and after Jennifer Doudna's lab, labs Fang Zhang and Broad Institute at MIT have discovered or made similar versions. What you can do over here is, there is a cousin of CRISPR-Cas9 called Cas12a that is slightly different than CRISPR-Cas9, and I'll tell you why. So in CRISPR-Cas9, you have a guide RNA, and that guide RNA allows cleavage of both the strands of DNA, and the Cas cannot cleave any other piece of DNA. However, what is so special about Cas12 is once the Cas12 binds to the desired piece of DNA and cleaves it, it can cleave any other single-stranded DNA that is present nearby. So what researchers have done is, let's say they have taken samples from cancer cells, from bacterial infection, from viral infection, and so on. What they have done is they have designed specific guide RNAs which will allow Cas12 to bind to that DNA of that, let's say, bacteria or virus. And after it has cleaved that piece of DNA from the host organism, it is provided a single-stranded DNA which is linked to a fluorophore. Because it is single-stranded and the Cas9 property that it can cleave any other single-stranded DNA in nearby vicinity, once it cleaves the single-stranded DNA, as you can see here, the fluorophore is released, and that is something you can monitor. This Cas12-based detection assay has also found a lot of implications or a lot of usage in COVID-19 diagnostics. And many of you might be aware of the Feluda test that just came out. Dr. Harshvardhan launched a few days ago that came out from a CSIR lab, IGIB in New Delhi. It is based on the same similar principle, wherein the COVID-19 DNA is bound by Cas12a, and then there is a fluorophore present, which once cleaved releases a fluorescence, which can be measured by any fluorescence detectable device, whether in the case of Feluda, it's a filter-based device, and it allows rapid diagnosis where you do not have very sophisticated scientific instrumentation available, and it's a mode of very fast and rapid diagnosis. So this is where it has found a lot of application in point-of-care diagnosis. Apart from that, where the field is evolving and where the field thinks that there is going to be huge implications is making precise changes in human DNA so that we can counter certain diseases. And so if you have to make changes in DNA, there are two kinds of changes that you can make. You can make changes in something we call somatic cells in humans. That means the changes made in those cells are not heritable. That means they are not going to be passed on to generations after generations. And then there are changes that you can make in the germ cells, such as the sperm, the egg, or the embryo, which are going to be passed on to generations. And one such somatic cell genome editing application that is in clinical trials now is for a disease that is well known as sickle cell anemia. So what happens in sickle cell anemia is normally you have, as you can see in a normal DNA, it's GAG. There is a single letter change. Instead of A, it's a T over here. So a single chemical change in the DNA leads to production of a protein that has a changed amino acid. So instead of glutamate, there is a valine present. And what happens in those cases is, rather than the round red blood cells that we see, the blood cells are somewhat sickle-shaped. The problem with these sickle-shaped cells is they are low in carrying oxygen, they clump together, and there are subsequent a number of problems leading to mortality in most cases. What people are trying to do with sickle cell anemia using CRISPR-Cas9 is they're trying to make this precise change over here, trying to change this T into A so that the phenotype can be reverted. This is one of the very many heritable disorders where people are trying to use CRISPR-Cas9 for editing. This part of the technology is in very advanced phases of clinical trials at this point. The other place where it has found tremendous amount of use in somatic cell genome editing is cancer immunotherapy. So everyone is aware of what cancer is. So one of the ways to counter cancer that has shown tremendous amount of promise, and people think that that is the way forward is using immunotherapy. That means modulating individuals' own cells so that they recognize cancer cells as foreign and kill them. And this is a recent, early, and this science paper published earlier this year showed remarkable phase 1 clinical results where they have used CRISPR-Cas9 to make certain knockouts of certain genes, as mentioned here, PDCD1, TRAC, and TRBC, and expressed, so these genes allow propagation of the cancer cells, and expressed NY-ESO1, which is a cell surface marker that specifically binds to cancer cells. So they have modified the T cells such that the individual's own T cells are going to recognize cancer cells as foreign cells and kill them. So this finding is an extremely important advance in therapeutic applications of gene editing, and highlights the potential of accelerated development of cell-based therapies using CRISPR-Cas9. In the final part, so the next part, so what I showed was somatic, means the changes can be made in the individual, and the individual will not be able to pass on to other generations moving forward. So this therapy is going to be somewhat personalized to the individual. Then there can be heritable changes that you can make using CRISPR-Cas9. And very early on, heritable changes were made in mice and pig. So as soon as Jennifer Doudna's and Emmanuel Charpentier's 2012 science paper came out, numerous labs started working on using CRISPR-Cas9 for various purposes. And one of the pioneering studies that came out in 2013 in Cell by Rudy Janish's group at MIT was he showed that you can use CRISPR-Cas9 to simultaneously knock out 5 genes in mice and have littermates or pups born out of the same mom at the same time have different coat colors. Moving forward, a few years later, it was also shown that using CRISPR-Cas9, you can remove certain locuses in pig that would make it suitable for xenotransplantation or organ donation in case a human wants certain organs from pig. So they made pigs that were suitable for xenotransplantation to humans. The moment these were made, the idea started floating about what about human embryos? Can they be modified? Can they be changed in ways that individuals want? And then came this whole hype of CRISPR babies. Can CRISPR, and in theory, CRISPR can be used. So can CRISPR be used to have babies that have high IQ or no baldness or a perfect voice or low risk to certain diseases, which we know that are, in certain cases, are heritable, or in certain cases, vision can be 20 by 20. Much like what we see in certain Hollywood movies where, okay, their designer humans are present. So yes, it is possible, but I'll come forward to what researchers have done to kind of not go forward on this path. While this all hype was going on, a Chinese scientist named He Jiankui, shown at the bottom, are the first studies of using CRISPR to make genetic changes in humans. His claims have later been unsubstantiated and unverified, and due to ethical considerations, he has been sent to jail subsequently. But this was one of the first reports, or the only report existent so far, where human embryos have been modified using CRISPR to have traits that otherwise would not be present and are desirable by individuals. So it paved the way to CRISPR babies having babies which can have traits that individuals desire. And this has been a hot topic of discussion, where people who have familial neurodegeneration want do not want their children to have it, so they want to have certain parts in those genes which are leading to, let's say, Alzheimer's or Parkinson's or ALS to be edited using CRISPR, so that the risk of their offsprings having those kind of diseases is reduced to a large extent. Given all these claims, scientists who were pioneering the field were concerned, and since then, scientists have come together and kind of arrived to a consensus that a prudent path should be taken forward for genome engineering, and particularly germline modifications. That means the traits that would be heritable from one generation to the other, because if they are not controlled, then a lot of illegal and unwarranted changes will be made to humans, much like having a good amount of muscles or having vision or things like that, or something we would call as superhumans, which will have tremendous impact on the society moving forward. Since the story came out about Chinese scientists, WHO has also stepped in and issued ethics about how human genome editing can be done, and Indian government has also come up with their own guidelines about what is allowed in terms of changes that you can make in human cells or human stem cells, and the changes you cannot make. So the guidelines are coming out and are rapidly evolving as the field is evolving. With that, I will close, and I hope I have not exceeded the time, and thank you all. So if I conclude, what I showed you is that CRISPR-Cas9 is an RNA-guided genome engineering toolbox with which you can tune it the way you want to make precise changes the way you want. The delivery and control are the key. The control is exerted at somewhat level by the PAM sequence that is present. Delivery has been a challenge. Some of the CRISPR-Cas9 has been delivered using viral vectors. In some cases, the proteins have been transfected inside cells. That is where some of the experimental challenge is still there, and a lot of basic researchers are trying to figure that out. However, even with the challenges that we face, there is tremendous amount of promise, both for biomedical applications for which there was no treatment and there still is no treatment, and tremendous amount of promise in agricultural innovation is still there, whether it's engineering crops that will be resistant to pests, that will be resistant to mold, resistant to drought, and so on and so forth. With that, I will close, and I will be happy to take any questions. Thank you all very much.
Thank you, Dr. Ashish. Now the forum is open for question. I guess there is no question, and okay. I think if Professor Murthy, can we conclude now?
Yeah, maybe people can put some questions in the chat box. Possibly there are people who are already putting. If you can see and then read out that, or maybe Ashish himself can see.
So Hale asks that there are some reports about off-target changes during CRISPR-Cas. Yes, there have been reports, and yes, that is one of the concerns that a lot of us have, and that is why now what people are trying to do is they are trying to find CRISPR-Cas9 with much higher specificity. So that's where the whole engineering of CRISPR-Cas9 is taking place. And one of the reasons that people have thought about is once you put the Cas inside the cell, and if it is constitutively active, there will be number of off-target effects. So what people are trying to do is trying to find ways that once CRISPR-Cas9 has done its job in the cell, is there a way to shut it down? So those are a couple of avenues that people are exploring at this point.
Ashish, this is similar to that people trying to use nanoparticles sometimes, okay,
Yeah.
for hyperthermia, for example. So the question that comes is, after it does its job, what is going to happen if the nanoparticles are going to
Sure.
stay back? Okay. So are they going to be further carcinogenic and things like that? So I think many other aspects associated with it has to be understood.
Yeah.
But it's a fascinating thing.
Yeah, the field has evolved in the last
up.
10 years and it is going to rapidly evolve in the next 5 years or so. Yeah.
At the same time, the way it looks like, it depends on in whose hands it falls. The same thing. I mean, starting from atomic energy onwards. Okay, science is science. It does its job, and it is the scientists who should try to utilize the science in the right perspective. So nanotechnology also, there's so much of things that one can do, chemical warfare one can do with nanotechnology. Similarly here also, as you are talking about trying to even tuning the DNA for the human beings, all that. I mean, we see so many Hollywood movies about this.
Right. This technology has initiated a big patenting war between UC Berkeley and Broad MIT, because there were two papers. So Jennifer Doudna's and Charpentier's paper came out, and within 6 months there were multiple papers from MIT coming out. And because of the promise of the technology, and there are so many startups privately held and publicly traded in US at this point, that there is a big patenting war going on.
How many groups in the country are working in this field?
So there are some people that are working, but everybody is taking on the application part. Like we use CRISPR-Cas9 to kind of change certain things in cancer cells.
Okay.
The basic research part is still mostly held by the folks who have discovered. So Jennifer Doudna and Feng Zhang at MIT, and those people are the ones that are leading it.
Thanks a lot, Ashish. It was wonderful. Thank you very much.
Thank you, Professor Moore. So can I leave, Professor Murthy?
Penmalar, you want to say anything? The last word as the HOD. You started it, you should end it. Are you there, Penmalar? Sorry, in such a case, let me propose a vote of thanks. Okay. So thank you very much, Ashish, for immediately accepting my request. We are soon going to have 2 talks on the physics Nobel Prize also next week.
That's very good.
We'll announce the dates very soon. This is very important that whenever we have these Nobel Prizes being announced, our students and faculty, if they come to know a little bit glimpse of what are the exciting things that are happening. So thanks a lot, Ashish, for that.
Thank you, Professor Murthy.
Telmalar for initiating this and seeing to it very much.
Thank you, Professor Murthy. Thanks. This was a wonderful opportunity. Thank you very
much. We'll wait for the Physics Nobel Prize talk next week.
Absolutely.
Okay. Saket is working on that. We will hear about it from him very soon. Thank you.
Sure. Thank you.